Assalamu alaikum dear students, hope all of you are fine. Let's start today's lecture. In the previous lecture we went through the dihybrid cross. I hope you got it. Dihybrid cross is a simply cross in which we are observing two pair of characters during the breeding experiment. When Mendel performed the cross, he chose two characters as shape of the seed and color of the seed, and the pure plants which he took for the process of monohybrid cross was round and yellow and wrinkled and green. Then he simply cross pollinated them and there was a formation of first filial generation, F1 generation. In F1 generation all the plants produced the seeds which were phenotypically round and showing color yellow. Then afterwards Mendel performed simply another step of the cross, that is he self pollinated F1 generation plants, and the final result were in the ratio 9:3:3:1. The 9 portions of the plants were having round and yellow seeds, 3 portions were having round and green seeds, and another 3 portions were having wrinkled and yellow seeds, and 1 portion of the plants showed wrinkled green seeds. Here again, like monohybrid cross, we observed that the dwarf recessive traits again expressed in the F2 generation. And when we see the genotypic ratio of the hybrid cross, it was like this: 1:2:1:2:4:2:1:2:1. When we go through this genotypic ratio, we observe that there is different genotype in few plants, there is the same genotype in few plant as that of the parent plant, and some plants also show the genotype which is seen in the F1 generation. But when finally Mendel performed the dihybrid cross, there was one of the most important conclusion from the dihybrid cross, and that conclusion was again simply used as law, and that conclusion is named as law of independent assortment. Law of independent assortment of traits. Law of independent assortment of traits, or you can say characters. It's one and the same thing. If you carefully observe the phenotypic ratio of dihybrid cross, we got 4 combinations. One of the combination was round and yellow, and another combination was round and green. Then third combination was wrinkled yellow, and the fourth combination was what we called as wrinkled green. Fine. Out of these 4 combinations, we are seeing that this is the same combination as that of the parent pure line plant. And if this combination is seen, it's exactly same like that of the parent pure line plant, but having recessive traits. These are the 2 combinations which are actually new: round and green and wrinkled and yellow. Both of these combinations are new, and this forms the basis for this law of independent assortment of traits, which states that there is no trait or character which is dependent upon another trait or character for moving from one generation to another generation. That means round trait or round character is not dependent always on the yellow character or yellow trait for simply inheritance. That means for moving from one generation to another generation. Yellow is not dependent on round, neither round is dependent on yellow. There is nothing like that. Or wrinkled, its character, it's not dependent, it's independent of the green. It don't depends upon the green character. That means wrinkled green, they are not independent upon each other. They can combine with any other. They can show any combination with other characters and move on from one generation to another generation. It's not like that. Round is always dependent on the yellow color and green color is always dependent on the wrinkled shape? No. There is any character that can combine with any other character and move from one generation to another generation. That was called as law of independent assortment, which is proven by these two new combinations. Because round, if it would have been dependent upon the yellow, then it would not have moved in F2 generation as round green seeds. And same, if yellow would have been dependent upon the round shade, then there won't be simply combination in F2 generation as wrinkled yellow, because it would only move from then previous generation to the next generation with only round. Same applies to wrinkled as well as green. Green is not dependent upon wrinkled shape, then only it will move from this generation to another generation during the process of reproduction. No, nor wrinkles is dependent upon the green, when there will be green and wrinkle combination and then only wrinkled character will move to the next generation. No, there is no any character that is dependent on another character for moving from one generation to another generation. This was called as the law of independent assortment, and it's one of the most important law which governs the genetics, especially the Mendelian genetics. And this was especially the result conclusion from dihybrid cross. That means from monohybrid cross we got three conclusions. First was the law of pair of unit factors, second law of purity of gametes or law of segregation of gametes, third law of dominance. All these three were concluded from monohybrid cross. Then the fourth one, that is law of independent assortment, had been concluded from the dihybrid cross. These four laws are collectively called as rules of inheritance or rules for inheritance. That means simply these are the laws, these are the rules which are simply followed when there is transmission of characters, transmission of resemblances, transmission of variations from one generation to another generation. If you remember, genetics is the study of simply heredity and variations, and heredity is transmission of characters from one generation to another generation, that is inheritance. During that process of inheritance, the rules that are followed by the characters from movement from one generation to another generation, all those rules are simply called as rules for inheritance or rules of inheritance, and which are usually four. That are first, law of pair of unit factors, law of segregation of gametes, law of dominance, and fourth one is law of independent assortment of traits. I hope this is clear to all of you. Now let's move forward. Now we are in the position, we are in the scenario where we can go through the wonderful world of genetic material, which we call as DNA, everybody, every one of you knows DNA is the genetic material present in human beings. Every one of you know it's just a biomolecule. It's also called as nucleic acid. But actually DNA is a biomolecule like its other biomolecules, carbohydrate, protein. DNA is also a biomolecule. Its full form is deoxyribose nucleic acid. DNA is called as deoxyribose nucleic acid. The DNA was first isolated by Frederick Mister. DNA was first isolated by Frederick Mister in 1869 from the pus cells. That means actually DNA was first isolated from nucleus of pus cells in 1869 by Frederick Mister. Because in cell we don't have only DNA, we have other molecules as well, and simply extracting just DNA from the cell, that is called as isolation, and that isolation was simply done by Frederick Mister in 1869 from nucleus of pus cells. After that, there were some experiments performed by the scientists, like there was a scientist Griffith He performed an experiment in simply 1928. Then there were another group of scientists called as Ivory, MacLeod and McCarthy in 1944. They performed experiments and showed that it's actually DNA which is transferred from one generation to another generation. In a cell, I repeat, in cell we have proteins, we have carbohydrates, we have fats and we have nucleic acids which we call as DNA or RNA. Even in our cells there is also RNA. But how you will prove that it's actually only and only DNA that is transferred from one generation to another generation through the process of reproduction? The proof was simply given by the scientists Griffith as well as Avery, MacLeod and McCarthy. They performed experiments in Griffith performed in 1928 and this group of scientists performed experiment in 1944 where they experimentally gave proof that it's actually DNA that is transmitted from one generation to another generation. Then finally there was a scientist couple which we call as Watson and Crick, which gave the structure of DNA, in 1962. This is the couple of scientists Watson and Crick which gave actual structure of DNA. What is DNA? What are the things which are present in DNA? What are the components of DNA? But actually Watson and Crick, before Watson and Crick, there are various scientists who tried to invent the structure, who tried to simply observe the structure of DNA, and using that data Watson and Crick finally gave the model of DNA regarding its structure, and they have been given Nobel Prize for the same, for giving the structure of DNA. Now you will see actually DNA is a polymer. According to Watson and Crick model of DNA, DNA is actually a polymer. Like protein is a polymer of amino acids. Protein is the polymer of amino acids. Same DNA is a polymer. DNA is a polymer of nucleotides. DNA is a polymer of nucleotides. That means DNA is actually simply formed by joining small units, and that small unit is nucleotides. Fine. For example, this is one of the nucleotide. It will join with another nucleotide like this, third one, fourth one, and it will form a long chain. That chain is called as polymer. Polymer is simply formed of monomer. Monomer means single unit. Polymer means multiple units. That single unit is DNA is nucleotide. That single unit of DNA is nucleotide. And when we observe the nucleotide itself is made up of three components. Nucleotide is itself made up of three components. One is nitrogen base. Fine. Another one is sugar, pentose sugar. And third component of nucleotide is simply what we call phosphate group. DNA is a polymer of nucleotides, and nucleotide itself is formed of three components. First, nitrogen base, second, pentose sugar, and phosphate group. Nitrogen base. What do we mean by the nitrogen base? Base means foundation, backbone, due to which DNA is formed. Why it is called as nitrogen? Because this possesses nitrogen atoms. It's made up of nitrogen atoms. Not only nitrogen atoms, there is carbon, oxygen, hydrogen as well. But nitrogen is necessarily present in nitrogen base. Nitrogen base is an organic compound, but it's a ring formed compound. It is a ring formed compound. Like if you have observed, we have different ring chain compounds in chemistry. One of the ring chain compound which we call as benzene, it's made up of carbon and hydrogen. Here we have simply ring chain organic compound which has nitrogen as component of its ring, and that ring chain organic compound is called as nitrogen base. And in nature, nitrogen bases are of two types. Nitrogen bases are of two types. One of the type is called as purines and another types are called as pyrimidines. One of the type of nitrogen bases are called as purines and another are called as pyrimidines. Purines and pyrimidines. Purine is a nitrogen base which is made up of two rings. Two rings. Purine is actually simply an organic compound but made up of two rings. It possesses two rings. But pyrimidine is simply a single ring organic compound. Single ring organic compound. And purines are of two types. Purines are of two types. One is called as adenine and another one is called as guanine. One is called as adenine and another one is called as guanine. I repeat, nitrogen base is organic compound which is in the form of ring. That ring possesses nitrogen as its component necessarily, that is why called as nitrogen base. The nitrogen base is of two types, purines and pyrimidines. Purines is double ringed nitrogen base and pyrimidine is single ring nitrogen base. And purine itself has two types, adenine and guanine. Fine. Then pyrimidine is of three types. We have thymine, and we have cytosine. We have cytosine and third one is uracil. All the three are single ringed nitrogen bases and named as thymine, cytosine and uracil. Fine. Then afterwards we have the second component which is called as pentose sugar, and third is phosphate group. But out of purines and pyrimidines, when a nucleotide is formed, there will be one purine or one pyrimidine used for the formation of nucleotide. For example, if you have to form a nucleotide, there will be use of only one nitrogenous base, adenine. Adenine represented by A, or you can use guanine if purines are used. If pyrimidines are used, again only one nitrogen base will be used for the formation of nucleotide, and that will be either thymine or cytosine Uracil is not used in DNA. Fine. When you have to form the nucleotide, you will join adenine with the pentose sugar and phosphate group. It will form nucleotide, one nucleotide. Then second nucleotide will be formed by simply you will take guanine, pentose sugar, phosphate group, second nucleotide. Fourth will be thymine combined with pentose sugar, phosphate group, and it will form third nucleotide. Same fourth nucleotide will be formed by cytosine, pentose sugar plus phosphate group. But in this way we will form different nucleotides. But again I am repeating, uracil is not used in the formation of DNA. I hope it's clear to you. Rest regarding pentose sugar and phosphate group we will discuss in the next lecture. Till then, stay safe, stay good. Thank you.